MCAT 2017 Cram, Biochemical and Biological Foundations of Living Systems, Passage 3, Cellular Effects of Metformin, a Drug for Diabetes Mellitus. As you view the reading of this passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these specific selections in order to answer the questions that follow. Good luck and happy reading. Paragraph 1. Type 2 diabetes mellitus is caused by a relative lack of or resistance to the protein insulin, which functions in maintaining normal blood sugar levels by facilitating transport of glucose into cells. Metformin is a common effective medication for type 2 diabetes mellitus. The drug has been shown to reduce rates of gluconeogenesis and increase uptake of glucose from the blood by skeletal muscles, but its specific mechanism of action is unknown. Paragraph 2. Researchers investigating metformin's mechanism of action isolated hepatocytes from rats. These are liver cells. They suspected metformin may interact with AMPK, a protein involved with a wide range of metabolic effects, such as increasing hepatic fatty acid oxidation, reducing hepatic glucose production, HGP for short, and reducing insulin secretion by beta cells of the pancreas. The researchers examined the activity of AMPK in hepatocytes treated with metformin, endogenous allosteric activator of AMPK or control solution. The cells treated with metformin were found to have increased AMPK activity compared to the control solution. The researchers performed a similar experiment in vitro with purified AMPK and its downstream substrate and found that metformin had no effect on AMPK activity. Figure 1. The effect of metformin on AMPK activity measured in hepatocytes, that's in vivo and in vitro. Hepatocytes were incubated for 1 hour with a buffered solution. AMP control or specified concentration of metformin. White control, gray AMP, black metformin. In vivo, in vitro. This is like in a petri dish or some test tube. This is like in a real specimen. P-value is less than 0.05 and p-value is less than 0.01. So these are significant. That means we can reject some sort of null hypothesis. Paragraph 3. The researchers also discovered an inhibitor called compound K that directly interacts with AMPK. Adding the inhibitor to hepatocytes incubated with metformin showed that compound K attenuates metformin's ability to reduce HGP. The results are shown here in Figure 2. Control, metformin only, metformin plus compound K, and this is the concentration of glucose. So the higher the concentration, the less the metformin activity, okay? Figure 2. Compound K inhibits metformin's usual effect of reducing HGP. Open squares, control, fill diamonds, metformin, fill circles, metformin and compound K. P-value less than 0.05 and p-value less than 0.01. So these are significant and we can reject the null hypothesis. Which statement about compound K is most likely to be true? Is it A, compound K lowers the Vmax of AMPK and is therefore a non-competitive inhibitor? Is it B, compound K decreases the rate at which AMPK binds substrate and is a non-competitive inhibitor? Is it C, compound K may cause hypoglycemia in a diabetic patient taking metformin? Or is it D, compound K increases blood insulin levels in a diabetic patient taking metformin? I'll give you a moment to think. This one may require some outside knowledge. All right. Us much about the biochemical properties of the inhibitor compound K. Compound K, we're told, lessens the activating effects of metformin on AMPK. Activated AMPK decreases hepatic glucose production. So if metformin is less capable of activating AMPK, blood glucose levels will rise, and higher blood glucose levels will trigger a greater release of insulin from the pancreas. So it's most likely true that compound K will increase blood insulin levels in a diabetic patient using metformin. All right, so the correct answer choice is answer choice D. For which reason did the researchers most likely choose hepatocytes, these are liver cells, as a target for metformin treatment? Is it A, the hepatocytes that were removed will regenerate in the livers of the rats? Is it B, to investigate the effects of metformin on gluconeogenesis? Is it C, to investigate the effects of metformin on insulin secretion? Or is it D, the effects of metformin are isolated to the liver? I'll give you a moment to think. In paragraph 1, the passage states that metformin, quote, increases the uptake of glucose by skeletal muscles. Therefore, we can infer that its effects are not isolated to the liver, so answer choice D is incorrect. Insulin secretion occurs in the beta cells, which is also mentioned in the passage. These cells are located in the pancreas. So, let's see, answer choice C is incorrect. Yeah, okay, so you wouldn't even need outside knowledge of insulin secretion by beta cells because it's stated in the passage. The main focus of the researchers' investigation is the effects of metformin on hepatic glucose production via the protein AMPK, okay? The researchers most likely chose to target the hepatocytes because hepatocytes increase blood glucose levels through gluconeogenesis. The correct answer choice is answer choice B. They wanted to investigate the effects of metformin on gluconeogenesis. All right, okay. Which of the following would be likely to increase blood glucose levels of a diabetic patient? Would it be A, metformin, B, compound K, C, AMP, or D, insulin? Now, this is basically just a restatement of a previous question. All right. Okay, so insulin's main function is basically to facilitate the transport of glucose into cells, thus reducing blood glucose levels. Okay. The protein AMP decreases hepatic glucose production via inhibiting the activation of gluconeogenic enzymes, phosphoenolpyruvate, PepsiK, and glucose 6-phosphatase. Okay. Therefore, a molecule that activates AMP will decrease blood glucose levels. So AMP and metformin both activate AMPK, while compound K is a direct inhibitor of AMPK. Therefore, compound K is likely to increase blood glucose levels of a diabetic patient. All right? Okay. Are possible explanations of why metformin increases AMPK activity in hepatocytes, but not in vitro. 1. Metformin acts on a protein upstream of AMPK. 2. An inhibitor of AMPK is present in the in vitro experiment. 3. Metformin changes the sensitivity of AMPK to phosphorylation. Or 4. Metformin changes the sensitivity of AMPK to phosphatases. Select all that apply. For this, you may need a bit of outside knowledge. All right. Within living cells, but not outside of cells in vitro in an experiment, tells us that metformin doesn't have a direct effect on AMPK. Metformin must require the presence of some other protein or compound to activate AMPK. I'm just going to show you the actual mechanism here, see if that's true, okay? It is unlikely, highly unlikely, that an inhibitor of AMPK is present because we're told that AMPK is purified, right? Often activates or deactivates an enzyme. Metformin could interact with AMPK to make it more sensitive to phosphorylation by another protein that is present, that is not present in vitro. Okay. Again, metformin could activate protein upstream of AMPK, which would go on to activate AMPK. An upstream protein would not be present in vitro. Things don't work out that perfectly in a test tube or a petri dish. The same reasoning that applies to antitrace 3 could apply to 4. Phosphatase is an enzyme that removes a phosphate group from its substrate, often causing deactivation of a protein. The addition of metformin may make AMPK a worse substrate for such an enzyme, but the same effect would not be observed in vitro. So, explanations 1, 3, and 4 are all possible explanations for why metformin affects AMPK activity in hepatocytes, but not in vitro. Right? Okay. How would starving the rats for 24 hours prior to experimentation affect the extracted hepatocytes? A, the hepatocytes would have an increased rate of glycolysis. B, the hepatocytes would have a reduced rate of gluconeogenesis. C, the hepatocytes would have an increased rate of glycogenolysis. Or D, the hepatocytes would have a reduced rate of lipolysis. You decide. I'll give you a moment to think. This may require outside knowledge. Okay, so Starvation takes away the rat's main source of glucose, okay, which is food, which causes them to produce glucose by other means. Glycolysis is the breakdown of glucose to pyruvate to produce ATP. This rate would definitely decrease during starvation to prevent the unnecessary use of glucose, okay? Gluconeogenesis, on the other hand, is the reverse of glycolysis, and it's basically the production of glucose from other sources due to the lack of glucose. So this is all seen here throughout the various organs, okay? And the rate would increase during our starvation, obviously, to produce more glucose. Lipolysis, okay, is the breakdown of fats. The rate of lipolysis would obviously increase during starvation. So this is wrong. And this is wrong as well. So A and D are wrong thus far. And so is B. So the default answer would be answer choice C. Glycogenolysis is the breakdown of the glycogen to the polymer to individual units of glucose. During starvation, the rat's hepatocytes would have an increased rate of glycogenolysis, okay? So basically, it's gluconeogenesis performed in the liver, where we break down glycogen stores, okay? All right.